Okay, so this is lesson 2 in genetics and molecular biology, and we are going to talk about single gene trait pedigree. So what I need you to do is turn to page 20 in your note packet, and we'll get started. So to talk about a single gene trait pedigree, we are going to use phenothiocarbamide, or PTC tasting. And the ability to taste PTC is considered a dominant trait. So how would you represent the dominant trait? If it's dominant, would it be big T or little t? Well, hopefully you said big T. And because it is considered a dominant trait, the ability to taste it, do you think that more individuals in the class would be tasters or non-tasters? Tasters or non-tasters? Hopefully you said tasters. And we're actually going to check that out on Friday. We're going to see who can taste it and who can't. We're going to fill in the table for PTC using the following answers. So these are our phenotypes, possible phenotypes, taster and non-taster. And these are our possible genotypes. We have homozygous dominant, heterozygous dominant, and homozygous recessive. So if we're a taster, and we know that that's a dominant trait, what would our genotypes be? Well, either big T, or big T, little t. Because if we're a non-taster, that means we have the recessive allele, and that means that both alleles have to be recessive in order for the trait to appear. Which genotype is heterozygous? Hetero means different, so that would be our big T, little t, and homozygous means the same, so big T, or little t, little t. So now we're going to look at a pedigree tree that represents a family and their ability to taste this PTC paper. So they're telling us that the circles are the females and that the squares are the males. And they're also telling us that the colored shapes are the homozygous recessive traits. So these are our non-tasters. Our non-tasters. And they want us to figure out what the other parents were in generation 1. And they're telling us that there are 2 generations. So here's generation 1, and this is their offspring. This is from the families. And then Mary and Bill have a family, and we want to know what their children would be. So this is basically family 1 over here, and this is family 2 over here. So we know that in order for the recessive trait to show, as it did in Mary, who's one of the offspring from this set of parents, that she had to have 2 recessive alleles. And the only way that she could get both recessive alleles was if this female was heterozygous. Let's see if that's true. So if I cross the recessive father with a heterozygous mother, I would get TT, there's your recessive, here's another recessive, and then heterozygous. So we only had 1 recessive female, which means that these 2 offspring both had to be heterozygous. Now, to prove that this female was not homozygous dominant, let me just do the crossover here for you. If we had that, then look at all of the offspring would be heterozygous. And that's not true, because we know that Mary, and then this child over here from this family, was also recessive for the trait, which means that this male had to be heterozygous. And that means that Bill and child 5 and child 7 also had to be heterozygous. So this was not a possibility. This parent, both of them, had to be heterozygous. So what possible genotypes could Bill and Mary's children have been? So now if Bill and Mary, so they want to know, this is the question mark, number 7. What is their children going to be if we cross heterozygous Bill with homozygous Mary? Well, that's this cross that I did over here. I'm going to get 50% recessive non-tasters and 50% dominant tasters. So their genotypes could either be big T, little t, or little t, little t. So that means that they're either going to be a taster or a non-taster. And I just said that 50% will be heterozygous, meaning a taster, and the other 50% will be a non-taster because they are homozygous recessive. Okay, so we're going to skip Do You Understand Pedigrees and Inheritance for right now, and we're going to flip over to page 21, and we're going to talk about Tay-Sachs real-world pedigree. Remember this is a defect on chromosome number 15. And let's look at here what's going on. They're telling you that the normal is clear, a carrier is half dark, half light, and then the affected is solid. Well, if you remember from what we talked about already about Tay-Sachs, chromosome number 15, it's a recessive trait. So this would be our Tay-Sachs, this would be our carrier, and then this would be our normal. Now, a carrier is going to appear normal, phenotypically appear normal. Genotypically, it will have the capital and the lowercase. Here, you don't have any trait whatsoever, so your genotype is pure and your phenotype is also normal. So let's see what happened when we crossed these parents. Okay, both grandparents, one was normal and one was a carrier. And when we cross them, you get 50% normal and you get 50% carrier. Now we know if we cross normal, we're going to get normal. So we're not interested in that. What they did then was took both heterozygous parents, which are these right here, big T, little t, and they crossed them. And let's see what we get when we cross them. You get normal, you get carrier, you get affected. Oops, I did that one wrong. You get a carrier and you get an affected. So that's exactly what we got down here. This one is normal. We got 2 carriers and we have 1 affected individual because it is a recessive trait. So let's now look at the different types of genetic inheritance possibilities. Most of what we've been talking about is this first one, autosomal recessive. That's the ability to taste PTC, that's a recessive trait, and Tay-Sachs, which affects the number 15 chromosome. That's also a recessive trait. So what does that mean, autosomal recessive? Well, it means that the gene is on an autosomal chromosome. And remember, autosomal chromosomes are our body chromosomes. So if you have the 2 recessive alleles, meaning little t, those are your alleles, then you inherit the trait. So that's like our PTC tasting, PTC and Tay-Sachs. We have not talked about any autosomal dominant disorders yet. The gene is still on an autosomal chromosome. But in this case, if you have one dominant allele, just one, then you inherit the trait. Sex-linked sounds exactly like what it is. The gene is on a sex chromosome. And in particular, the X. Men are more likely to have the trait because they have only one X chromosome. For women, the second X will usually dominate. So what does that mean? Well, it means that if a woman has one copy of the mutation on one of her X chromosomes, then she typically will not have she will not express the trait. It will not phenotypically be seen because her other X, which is dominant, will mask it. But men, on the other hand, will probably have the trait because they only have one X. Their other one's a Y. So if it's present on the X, then they usually display it. Random mutation means exactly what it sounds like. It occurs at random, and it is not inherited. Okay, that's it. I will see you in class Friday. Make sure you get all your work done. Please see the updates on Schoology.